Hi, listeners. My name is Stephanie Hare, and I am a postdoc in the Department of Chemical Engineering at the University of Washington. I'm going to talk to you all today about a project that I've been working on, probing the effects of electrostatics on silica oligomerization. Materials generated from silica have a variety of useful applications in the realms of drug discovery, energy storage, even synthetic bone. But I'm showing diatomaceous earth here because it's a ubiquitous substance that you can find at a variety of retailers. And you may be familiar with this substance if you've ever had to deal with pests, because that's what it's primarily marketed for. But it's made of these organisms called marine diatoms that have these really interesting exoskeletons and really interesting morphologies that are made out of silica. And you can see some of those here. So this is on the nano to micrometer scale. And this reaction to generate silica into these interesting morphologies occurs on a protein called silafin, which is a portmanteau of silicon and affinity. So these are proteins that have an affinity for silicon. And they're able to template the oligomerization of silica in order to make these interesting structures. And even beyond that, if you were to extract silafin from these organisms and take from that even just a piece of it, so just a small peptide, and the peptide that I'm going to be talking about in this slide is called NatSil1A. If you take that peptide and put it in a solution of orthosilicic acid, you can actually precipitate silica nanospheres on the order of seconds to minutes. So what I'm showing here from A to D is a time lapse of 8 minutes of NatSil1A added to a buffered orthosilicic acid solution. And so you can see what orthosilicic acid is right here. It's a very simple structure, and it's just sequestered from solution and oligomerizes when you have these peptides present. But an important point is that if you just have a solution of orthosilicic acid, it doesn't spontaneously oligomerize in order to make these structures. So the peptide or a protein is necessary in order to catalyze this reaction to actually happen. So we're wondering, one of our questions is what the silafin protein or the peptides that make up silafin do in order to catalyze this reaction. How does it actually work? So I'm showing here a picture of NatSil1A, and this is a 15-mer peptide. So we have the primary sequence of amino acids here. And then we also have several post-translational modifications. So we have all of these phosphate groups, those happen post-translation. And we also have these long polyamine chains. And we can think of this structure as a collection of positive and negative charges. And our working understanding of how this peptide actually works in order to template this reaction is that having these peptides in solution, the conformation of these peptides changes and they actually aggregate into these globular structures on which we think this silica oligomerization reaction occurs. And so this is our working understanding of how these peptides are able to catalyze this reaction, but we still have a lot of questions about how this actually works and how we might be able to manipulate this reaction in order to make materials in the lab. And so our current research questions are, how does the interfacial structure of the peptide aggregate template the morphology of these structures? So how do we actually get the correct shapes of these structures? And this could, like I said, help us with biomimetics or biomimicry so that we can actually, or someone can make these structures in the lab. And we're also wondering, what role does the peptide specifically play in this reaction? And again, specifically, how do the electrostatics of the peptide come into play in this reaction? And can we actually emulate the role of the peptide using a simpler model? So instead of modeling an entire peptide aggregate, actually just using individual amino acid residue analogs or having a charged interface in the vicinity of this reaction. And then the smallest scope of all in terms of questions, what is the mechanism of this silica oligomerization reaction? And we're wondering about this mechanism in the gas phase, modeling it in the gas phase, in solution, and with a peptide present. And so what I'm going to talk about today is mostly the mechanism that we found for the silica oligomerization reaction under slightly different conditions. And I'm going to touch on, hopefully, what this means, the role of the peptide in catalyzing this reaction. And so to briefly introduce the types of calculations that I do, I'm actually a computational chemist, and I primarily work with DFT calculations and calculating stationary points along reaction networks. And so if you've taken introductory chemistry, you've probably seen a plot like this before. It's a reaction coordinate diagram where we have reaction coordinate versus potential energy. And on this diagram, at an energy minimum, we have a reactant that goes over a transition state structure at an energy maximum and then goes to a product. And this activation barrier is what we can use. If in simple cases, we can use something like the AIChE equation in order to calculate a rate constant and ultimately calculate a rate. And so what I actually do is I take the stationary points along this reaction coordinate, so the minima and maximum, and optimize those structures at some level of theory, and then calculate the energy at the same level of theory or a different one, and then use that in order to make conclusions about how this barrier affects the rates of this particular reaction. And a lot of the time we have competing pathways from reactant to several different products or even the same product. And if we have these competing pathways, then what we care about is the difference in energy between these transition state structures. And again, in very simple cases, we can use this to calculate a product ratio. But most of the time we have to use more complicated rate theories in order to actually calculate realistic rates. But I'm just showing this as an illustration to explain the conclusions that I draw later in this talk. And so what I've been able to map out, based on structures that actually were already mapped out in the literature, was this silica dimerization network. So we're just looking at the first step toward silica oligomerization. Of course, this would be repeated in order to make longer structures, but there are a few possible paths for the reactant complex to generate a silica dimer. So what I'm showing here is the reactant complex. We have an orthosilicic acid molecule and a deprotonated orthosilicic acid molecule, and then some catalyst, which I'll talk about a little bit later. But what happens in this pathway of the reaction network is that the reactant complex goes over a transition state structure where this deprotonated oxygen, this oxide, attacks the silicon center of the other orthosilicic acid molecule and generates this intermediate structure which has a pentacoordinate silicon. And this intermediate structure can then go over another transition state structure where it loses a hydroxide and simultaneously deprotonates an adjacent hydroxyl group. And we end up with this deprotonated dimer of silica with a water molecule that I'm calling the product complex. So this is one path that can be gone down. There's actually another path where intermediate 1 is able to isomerize to a second intermediate 2. And I'm showing here the difference between intermediate 1 and 2. They're super similar, but with intermediate 1, we have a hydroxyl group that is coaxial within this internal oxygen of the intermediate, whereas in intermediate 2, we actually have 2 coaxial OH groups. And in this part of the pathway, intermediate 2 can go over a transition state structure where this top hydroxide leaves, simultaneously deprotonating an adjacent hydroxyl group to generate the product complex again. But actually, there's a second possible pathway starting from intermediate 2 again. So this bottom hydroxyl group can leave and deprotonate a hydroxyl group from the other silicon of the intermediate in order to generate the final product complex. So this is a relatively complex reaction network. And I haven't even talked about the possible catalyst yet. So what this AIChE group can be, it has to be a catalyst that's able to donate a proton. So we can either have no catalyst if we're talking about the inherent reaction where it's not catalyzed at all, or we can have a water molecule, or we can have one of 2 protonated amine molecules. We can have methylammonium ion or guanidinium ion. And I'm saying that we can have these things as in I have calculated the energetics along the reaction networks for all of these 4 systems to see how they compare and to see why in solution with no peptide present, possibly why we don't see formation of these silica oligomers. And so when I mapped out the energetics of these reaction networks for all four systems, they're quite complicated. So in the inherent system, we have the three possible pathways that I showed. Intermediate one can either go over TS2 or this isomerization transition state structure, and then intermediate two can go over one of two possible transition states to get to the product. But in the case of having a catalyst, it gets even more complicated because the catalyst itself can either be donating a proton and acting as a true catalyst, or it can be just acting as a spectator, and all of the proton transfers are done internally. And so that's why you're seeing more complex reaction networks when there's a catalyst involved. And to simplify things a little bit more, I'm just going to show the lowest energy pathways for each of these systems and compare those. And the interesting thing that we see in this case is that the lowest energy pathway, when we don't have a catalyst or we only have water behaving as a catalyst, is different than if we have a protonated amine acting as a catalyst. So in the case of no catalyst or water, the lowest energy pathways are going through this isomerization process in order to get to the final product complex. Whereas in the case of a protonated amine, the energy of the relevant transition states are lowered enough that the lowest energy pathway actually becomes this direct pathway from intermediate one to the product complex. And so we thought this was an interesting conclusion that ultimately we found using this level of theory and using this very simple model, that a protonated amine catalyst appears to change the lowest energy pathway from reactant to product complex in the silica dimerization reaction. And so again, when we have no catalyst or a water molecule as a catalyst, the lowest energy pathway is this isomerization pathway, and the direct pathway from intermediate one to the product is a secondary pathway, which probably is still accessible at room temperature, but is not the lowest energy pathway. But then if the catalyst is a protonated amine, the direct pathway ends up being the lowest energy pathway. But again, this isomerization pathway is still probably accessible at room temperature. And so in the future, what we have to do is, in order to calculate an actual rate of production of these products in each of these cases, we need to do a microkinetic model and incorporate all of these possible pathways and their relevant barriers in order to get a reasonable estimate for the rate of these reactions. And another next step that we want to be able to do is something called a degree of rate control analysis. So this was started by Charlie Campbell at the University of Washington. And it's basically an analysis to determine which species along a reaction pathway change the rate the most. And it can give us an idea of what the catalyst, what the peptide is doing in solution in order to catalyze this reaction. We also are looking at the next phase of this project being molecular dynamics simulations in the condensed phase. So going back to this picture of the peptide aggregate and thinking about the charges of the peptide aggregate, we're wondering if maybe we can model this in a simple way, obviously going more complex than the calculations I've done so far. So doing MD in the condensed phase, but modeling the electrostatics of this peptide aggregate as just an interface with no mass. And since we probably don't actually have a good model for the specific electrostatic potential of this peptide aggregate, we're going to have a simple, a more simple model for these electrostatics. And so what we're proposing doing first, just trying it out, is a very simple pattern of alternating positive and negative charges or positive and neutral charges, and using that at the interface of air and water, and this water being a solution of orthosilicic acid, where we're using a reactive potential in order to be able to observe these reactions in solutions. And so this is just kind of an idea that we're playing with, but it will be the next step for this project. And so with that, I'd like to acknowledge my group, especially Jim, my boss, as well as the entire group. They're awesome. And also the University of Washington and the Department of Energy for funding. So thank you all so much for listening.